Welcome back. So we are going to start the second part of the program. The first speaker is Daria Sokolova from Spanish River. She's in the biotechnology program. So when she came here, she already had a lot of experience with pipetting and doing experiments in the lab. And actually already know her name from a science fair because, right, you are one of the finalists for the science fair, the whole state of Florida. You got the fourth place. It's a great achievement. And so I was very excited for Daria to be here and to really put to test her experience at school with how it is to be in a real lab. And she did great. And from what the other interns told me, so you know, right, that I always have a ping pong challenge going on. And apparently Jake was the one that started already like a good ping pong, but Daria was our underdog that just came strong and won the ping pong challenge of the interns. So, good job on the ping pong. Let's see what you did in the lab.
Good morning. My name is Daria Sokolova, and I'm a rising senior at Spanish River High School in the Biotechnology Academy. This summer, I had the incredible opportunity of working at the Bolton Lab, which focuses on the effects of genetic mutations on neural circuits, as they are modified in neuropsychiatric disorders. My project specifically is centered around analyzing amygdalar axon morphology in the prefrontal cortex of the TBR1 mouse model of autism spectrum disorder. Autism spectrum disorder is a neuropsychiatric disorder most commonly categorized into three main behavioral characteristics: social communication deficits, language deficits, and restrictive and repetitive behaviors. Autism is caused by genetic mutations or contributing environmental factors, those being maternal immune activation or prenatal exposure to toxins. And of course, autism spectrum disorder is an umbrella term, meaning it possesses a wide range of clinical heterogeneity, and each case is completely unique. This diagram highlights this, including the variety of comorbidities such as anxiety, ADHD, depression, and cognition deficits that can be present along this neuropsychiatric disorder. In terms of human neuropathology of ASD, or in other words, the morphological alterations seen throughout the brain in autism, the commonality between all deficits is the modification, formation, function, and maintenance of synapses throughout the brain. If looked at more specifically, the cortical area, visualized on the left-hand side, is responsible for higher-order brain functions, like memory, sensation, and perception. This area can experience neuronal migration and maturation modifications in autism spectrum disorder. The subcortical area, which is visualized on the right-hand side, is mostly responsible for learning, memory, and fear conditioning. This area can experience abnormalities in specific subcortical pathways. The amygdala is a part of the subcortical area that I analyzed in my project. It is responsible for receiving and sending signals throughout the brain. Together, these two functions, parts of the brain, work together to maintain proper brain activity throughout the brain. More specifically, my project deals with the amygdala, a part of the brain vital for fear conditioning and emotional learning. It is visualized in the orange circle on the diagram, along with its reciprocal connections to and from the prefrontal cortex, a part of the cortical area responsible for cognitive control, memory, and planning, and executes behavioral aspects of the amygdala as well as other brain areas. When acknowledging the behavioral characteristics of autism spectrum disorder, the roles of these brain regions makes it clear that both the amygdala and the prefrontal cortex play a significant role in ASD modifications. To highlight the importance of these two structures, researchers have conducted a variety of human-based experiments that justify their functional alterations in terms of ASD. These images represent a few of the human studies I looked at which exemplify reduced neuronal activation in individuals with ASD. In the leftmost study, a study utilizing fMRI imaging was used to compare neuronal activation when study participants were presented with neutral faces. As shown, there is a clear difference between the activation in the amygdala of the comparison group and that of the ASD group. The image on the top side of the left-hand image shows clear neuronal activation of the amygdala, while this bottom image shows reduced and limited activation of the amygdala. Similarly, the study on the right was conducted between patients of familiar faces in which they recognized the prefrontal cortex activation. Similarly to the left-hand side image, there was reduced or limited prefrontal cortex activation in this area. Combined, these two studies not only indicate the significance of the prefrontal cortex and the amygdala in relationship to autism, but also identify the functional commonality between mice and humans in relationship to studying this disorder. TBR1 as an ASD mouse model. TBR1, or T-brain1, is a neuron-specific transcription factor. A transcription factor is a protein that binds to a promoter to ultimately express the expression of genes. This transcription factor is crucial in the development of the cerebral cortex and the amygdala, while also playing a big role in the development of the cerebral cortex and neuronal migration and axonal projections within the brain. However, my project specifically utilized a TBR1 haploinsufficient model, meaning one of the genes in the TBR1 was knocked out. De novo and inherited TBR1 mutations were both found in patients with ASD, making this gene incredibly important to study as a model. Furthermore, haploinsufficiency of TBR1 has not only shown cognitive abnormalities and impaired brain wiring, but also a variety of behavior tests in mice have put its place as a high-confidence risk gene when studying this disorder. These two images further exemplify this, where it shows the connection from the amygdala into other parts of the brain. In the wild type or normal mouse, there is a clear connection of fiber bundles from the amygdala, as shown in this image. In the TBR1 haploinsufficient sample, that connection is simply not there. You can appreciate the absence of a fiber bundle connecting the amygdala, suggesting a specific sensitivity of amygdala neurons to a TBR1 mutation, and this ultimately motivated us to investigate its connection to the prefrontal cortex. The procedure for my project began with a viral injection of a GFP-based chernodopsin marker into the amygdala. This was performed by my mentor, Ingo. Chernodopsin is a light-activated ion channel protein that, when paired with GFP in light of a specific wavelength, it works together to indicate the presence of axonal projections into the prefrontal cortex. This process is also commonly referred to as optogenetics. Then 3 weeks was allowed for expression of the viral injection, followed by perfusion and fixation of the brain. Then I completed slicing and immunostaining, where I immunostained for the presynaptic protein DGLUT1 and the postsynaptic protein Homer1. This, combined with tissue mounting, ultimately resulted in a slide ready for confocal microscope imaging. Over the past 6 weeks, I successfully learned how to use the Zeiss LSM980 confocal microscope to produce these images and ultimately gain a valid conclusion for my experiment. Finally, the last part of my project was analysis on ImageJ and GraphPad. ImageJ is an image processing software commonly used among scientists to study and analyze microscope images, and GraphPad is a data processing software. Here, the top image is an example of an epi wide-field overview of one slide, which usually contains about 10 to 12 brain slices 50 microns in thickness. The true expression of the GFP and the marker can be seen by the brighter green sections near the midline of the brain. The bottom image is a zoomed-in version of one of the slices that focuses more on the expression in the prefrontal cortex. The bright light seen in this middle compartment right here represents the axon bundles projected from the amygdala. Over the course of the 6 weeks, I collected data from over 20 samples and 100 slides in total. To measure the innervation of amygdala axons into the prefrontal cortex, I utilized an identical rectangular region of interest, or ROI, shown in this image. Aligned with the midline of the brain, ImageJ then calculated the brightness intensity of the fluorescence that represented the presence of axon densities, or in other words, axon bundles in the prefrontal cortex. This graph compares the brightness intensity of the fluorescence between the axon bundles of wild type, shown by this black bar, and TBR1, shown by this blue bar. There is a significant decrease in axon density of the TBR1 mutated side, indicating that mice with ASD-like behavior and morphology show decreased axonal density in the prefrontal cortex. This graph on the right is a similar representation, where the black bar is the wild-type samples and the blue bar is the TBR1 mutated samples. However, this graph shows the brightness of individual pixels from left to right on the ROI. And as you can see, this peak shows a significant decrease from the wild-type to the TBR1 mutated samples. One thing that I found really interesting in this particular plot is that the first area shows really low intensity. This is actually representative of the layers of the brain, which can be indicated in this image right here. So the first section is low because this is the midline of the brain. This high peak is representative of the expression of the GFP and indicates the presence of axon bundles from the amygdala into the prefrontal cortex. Finally, the graph levels off because there is no more expression in that gene. Ultimately, I found that TBR1 haploinsufficient mice show significantly decreased amygdalar axon densities in the prefrontal cortex. Combined with the previously proven information of the importance of the amygdala and the prefrontal cortex in executing cognitive functions, with my conclusion and additional studies regarding more genes in other parts of the brain, we can ultimately promise that there might be a development of treatments for ASD. Lastly, I want to say a big thank you to everyone who allowed me to have this amazing experience. To Ingo Asbeck, who was an incredible mentor and helped me learn so much in this short amount of time. The entire Bolton laboratory, specifically Dr. Bolton, for all her amazing guidance throughout my experience. Ilaria, for all of your help throughout my internship. MPFI supporters who made this experience possible in the first place. MPFI core facilities, and of course, all of the interns who made this experience fun. Thank you so much.
Questions for Daria?
You had a descriptor of a mouse about 2 or 3 slides back. Mm-hmm. And it's a term I was totally unfamiliar with. Is it a special, I know that we have special animals here that we do lab work on, but I've not heard that term before. And I don't know how to take it back in your slides.
Yeah, Kevin, can you put the slides back so Daria, you can go back to the slide?
Yeah, keep going. Forward, yeah.
This one?
Let's see. I'll have to ask later. It's, no, it's after that. But
is it TBR?
It was a particular kind of, I guess, mouse, and I just didn't understand it. But we'll talk about it on break. Thank you.
Any other question? And then Alana.
Hi, Daria. Quick question about your microscope images that you took.
Yes.
So you said you compared the 2 different types, so basically the wild type and the one that you were examining. Can you say how many mice or how many slices of each you compared? And what was the hardest thing to get kind of the regions and all the other stuff to match up between the different imaging that you did?
So the question was how many mice I used in total and how many slices I used for each sample. For mice, I used 20 mice in total, so 10 for wild type and 10 for TBR1 mutated samples. And so each mouse was representative of 1 slide, so there was about 10 slices of the brain of 1 mouse in that 1 slide. And then I compared the brightness of the TBR1 mutated with the wild type of the GXP expression. In terms of your other question, with the specific area of the brain that I used, the prefrontal cortex, the ROI was within the prefrontal cortex, specifically the prelimbic area of the brain.
And we have another question for Alana, right? So, yeah.
So you showed us one of your slides where you had a lot of different steps that you went through for the entire project. What would you think was the biggest challenge you faced?
I think the biggest challenge, there were several challenges, and although I didn't complete the viral injection myself, it was often difficult to get the expression directly in the prefrontal cortex from the amygdala. So several of the samples I could not use for my project because of that incorrect expression, but the viral injection is a very difficult process. And in addition, I didn't really know how my data would turn out until I really did the data analysis. So I wasn't really sure until up to 2 weeks ago that it was actually significant what I did. So that was definitely one of the hardest aspects of my project.
That is real science, by the way. You work a lot and then you have to wait. Thank you so much.